Welcome. My name is Yulong Bai. Today, I would like to share with you our recently published work, scMAPA. Many messenger RNA contain multiple polydenylation sites in their 3' untranslated region. With these different PA sites, alternative polyadenylation, short as APA, produces transcripts with various 3' lengths from the same gene. We call them APA isoforms. Since there are many regulatory elements located in 3' UTR, these APA events can affect multiple post-transcriptional regulation mechanisms and consequently function in diverse cellular processes. Single-cell RNA-seq data provides a good opportunity to profile the APA events in a cell-type-specific manner. To achieve this goal, we developed a bioinformatics tool named scMAPA. Here are major features of our tool. First, scMAPA is compatible with both 3' captured and full-length single-cell RNA-seq data. Second, scMAPA can adjust for undesired sources of variation, such as the sample origins, if samples were extracted from different tissues. Third, in both simulation and real data, scMAPA outperforms existing methods in sensitivity, robustness, and stability. Next, I will briefly walk you through the major steps of scMAPA algorithms. If the input is 3' captured reads, scMAPA first passes the short reads to the start point of 3' UTR based on references, so that the read coverage of 3' captured library, like this bell-shaped curve, will be transformed into cumulative curve as shown here. Next, we extended a well-known APA quantification tool, DARPAS, which was originally designed for bulk RNA-seq data to detect polyadenylation sets from transformed 3' UTR signal. Basically, it solves a change point detection problem formulated as this equation by quadratic programming to minimize the difference between the observed read coverage and the expected read coverage. It estimated the location of PA sites and quantified APA isoforms simultaneously. Then, scMAPA models the abundance of APA isoforms against the cell type and other covariates through a multinomial logistic regression. To benchmark the performance of scMAPA using the ground truth, we developed a novel simulation pipeline. In this pipeline, we simulate the conventional single-cell RNA matrix by splatter and APA isoform ratio matrix based on a parameter named SD isoprop. SD isoprop is defined as the standard deviation of the proportion of APA isoforms across cell types. We found this measurement can effectively distinguish APA genes from non-APA genes, as shown in this density plot. Then, by multiplying these two matrices, we got the APA isoform specific matrix, which is the simulation data for benchmarking. In the results, as shown in this figure, across different simulation scenarios, scMAPA consistently achieved a higher sensitivity while having a similar specificity. Then we assess the PA site identification accuracy of scMAPA and other methods by running these tools on human PBMC datasets downloaded from 10x example data repertoire. From the line plot on the left, we can see scMAPA achieves the highest PA site identification accuracy. Also, since 1,000 and 10,000 cells datasets were derived from same healthy donor and 5,000 data from another healthy donor, we expected that the APA profile should be similar in these three datasets. As you can see in this bar plot, scMAPA identified the most common APA genes across three datasets compared to other methods. This suggests that scMAPA is more robust when sample size vary. Next, to demonstrate how the cell type specific APA profiles enables us to understand the post-transcriptional regulation, we analyzed the mouse brain single-cell RNA-seq data. We first identified significant APA genes across cell types and estimate the effect size of these APA genes, which is then visualized as this heat map. Based on the estimated effect size, we calculated the Euclidean distance between the cell types and performed the PCA analysis. Well, both analysis supports the previous findings that immune cells and neurons are the most different in terms of APA profile. We further revealed that immune cells are most different from all the other cell types. Next, using the same data, we demonstrated that scMAPA can adjust for undesired source of variants to uncover APA functions. In mouse brain data, cells were collected from multiple brain regions. As shown in the t-SNE plots based on the expression values, some cell types distributed evenly across brain regions, but in astrocytes and neurons, there are clear separation by brain region. So we suppose that APA genes associated with brain region could also be mistakenly identified as cell type-specific APA gene. To see if scMAPA can adjust for such impact, we compare the results from 2 runs of scMAPA, with or without brain region adjusted. In total, there were 113 genes that were not identified in brain region adjusted scMAPA. These APA genes are expected to be related to the brain region. Indeed, we observed that the human homologs of these APA genes are significantly upregulated in brain cortex compared to other brain regions in GTEx data. In conclusion, we developed scMAPA that does not rely on certain assumptions of density and shapes of 3' captured rates. scMAPA outperforms existing methods in accurately and robustly identifying APA genes. scMAPA identifies APA genes specific to each cell type in a statistically rigorous model that allows controlling for undesired source of variation. This can help to elucidate the function of APA in a complex system. For more details, please check our paper through this link. Thank you.